Hello and welcome to Nikolay's genetics lessons, and today you are lucky because I'm not going to solve any problems but going to just concentrate on two conceptual ideas: what is a broad sense heritability versus narrow sense heritability, what's the difference, because this is sometimes hard topic for many students to understand, and especially for those people who just trying to learn breeding and selection on their own, and they have trouble understanding the difference between broad sense heritability and narrow sense heritability. So today, under I hope few minutes, I will try to explain you the difference so you would know once and forever. So let's start with the formula itself. For example, broad sense heritability would be H capital squared, which equal to variance genetic divided by variance phenotypic. So this is simple formula, and here is another formula for narrow sense heritability, which is H small squared, which is equal variance additive divided by variance phenotypic. Now let's return to the first formula, and basically here I can say variance total genetic divided by variance total phenotypic, because variance genetic also consists of variance genetic consists of three components. The first one would be variance due to additive traits plus variance that is going to be due to dominance plus variance epistatic. So let's talk about each one. So what is a variance additive? Imagine that we have one locus with one gene and two alleles, dominant allele A and recessive allele A. And let's say we cross two heterozygous genotypes, that is going to be heterozygous for this particular gene. And because this is additive trait, let's say dominant allele A would add 2 centimeters, let's say this is plant, and recessive allele A would add 1 centimeter to the height of the plant. So this is additive trait. Now let's say we make Punnett square, and let's see what results such a cross produce in the next generation. Once again, dominant allele at 2 centimeters and recessive 1 centimeter. So here we are going to have homozygous dominant genotype, heterozygous, and homozygous recessive. And we are going to have here 2 and 2 centimeters, 2 and 1, 2 and 1, and 1 and 1. So we are going to have here 3 distinctive genotypes. This one would make 4 centimeter plant, and this one would produce plant that is going to be 3 centimeters, and this one would produce a plant that is going to be 2 centimeters. So such traits we call additive traits. And by the way, most of traits in any organism, whether it is plant or animal, is additive by nature. Consider, for example, such trait as height or body weight. Such traits would be additive, because hundreds, if not thousands of genes would participate and would add a little bit to your genotype, whether you're going to be tall or short. And usually parents who are tall have also tall children. It is very unlikely that they would have children who is going to be very small. Now let's talk about, so we talk about what is a additive trait. Now let's talk about trait that is dominance trait. Once again, the same picture. We have two parents who is heterozygous, both heterozygous. And now let's build a Punnett square again. And we are going to see almost the same picture. The same genotypes, 3 genotypes, but only 2 phenotypes, because this 3 would make one dominant phenotype, and this one would make recessive phenotype. So this is a dominant trait, and example would be ABO blood group system, where 2 alleles A and B are codominant and allele O is recessive. But not many traits would be due to dominance, but still such traits do exist. And now let's talk about the last component of the genetic variance, which is variance due to epistasis. And now let's take a look at this picture. So basically what we see here 23 pairs of chromosomes. Now imagine that we have 2 genes. Let's say one gene would be on the chromosome number 1 in this locus, and another gene would be on the chromosome number 11 somewhere here, and let's say this is gene A and locus A, and this is gene B in locus B. And no matter whether we have here dominant allele or recessive allele, actually this trait would be expressed only if here, for example, we would have dominant allele. Then this trait would be expressed, this allele would be expressed. Or if we have recessive allele here, no matter whether this dominant allele or not, it's not going to be expressed. We can say that this gene would be epistatic to this gene, and we can say that this works like molecular switch. So it can be whether on or off, and it would affect how this gene would be expressed. So this is an example of the variance due to epistasis. So when we talk here, once again, we are talking about variance genetic, or we can say variance total genetic divided by variance total phenotypic. And once again, so let me put arrow here, and now let's talk what is a variance phenotypic. Basically, variance phenotypic would be variance genetic plus variance environmental, variance due to environment. So do not mistake it with variance due to epistasis. So E here stands for the variance environmental, or we can capitalize this letter, so you would see that this is 2 different letters. So basically, once again, variance total genetic divided by variance total phenotypic would give us broad sense heritability. Now, as you see, narrow sense heritability is when variance additive, which is example here, would be divided by variance phenotypic. And basically what we can get. We can get any number here between 0 and 1. Now let's talk about these numbers. The bigger number would be to 1, or we also can say to 100%. So 1 would equal to 100% and 0 would also equal to 0%. The greater number we can get here, that is, would be closer to 1, the greater would be additive trait in phenotypic trait. So the greater would be this component. And sometimes we even can get 100%. And example would be, once again, ABO blood group system where environment doesn't have any effect. So variance phenotypic would equal to variance genetic, because environment doesn't have any effect on ABO blood group system. Only genetic composition would affect whether you would have blood group A, B, AB, or blood group O. So, for example, if you got 0.75 here, what this number would tell us? This would tell us that this is this plant or group of plants or animals have a good can be good candidates for selection, breeding, for breeding and other purposes, because variance genetic, variance additive here is important component. But sometimes we can see numbers that is going to be very close to 0, for example 0.1 or even 0.01. That means that all the variance here would be due to variance environmental. Sometimes we can see it in inbred animals, even 100% inbred animals, like some laboratory mice, can still be slightly different. They may have slightly different body weight, may have different other phenotypic traits, but all those traits would be due to just environmental factors. Some mice may get some disease and wouldn't be fed well. Others may avoid this disease, would feed better, and would gain more weight. So in this case, of course, if we will find that our population would have a very small number here, we cannot use such population for selection breeding, because all the population would be genetically similar. So that's why it is important to find this number. Now let's talk about what is the difference between this broad sense heritability and narrow sense heritability. As I said, most important factor here in variance genetic would be variance additive. Most our variance and most of our traits would be due to additive trait. As I said before, this can be how tall we are, our body weight, size of the corona of the plant, also distance or which we call internode between leaves, and many other factors would be mostly due to variance additive. That's why we have a separate formula where we find what is the proportion of the variance additive to total variance or variance phenotypic. One more time. Basically, here we are saying that trait is due to small effects of many genes. Here we are talking about few genes that would affect the trait, and here we are talking about genetic switch that would affect how other genes would be expressed. Now I hope you understand the difference between broad sense heritability and narrow sense heritability. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates and see you in the next video. Goodbye.